play currently within CNS clinical trials? And more specifically, what's the problem? Because there are quite a few problems within the clinical research area of CNS. And the primary problem is that CNS studies fail, and they fail quite a bit. Why is this? Well, first of all, there's a limited understanding of the disease pathophysiology in many cases and in many indications within neuroscience. And additionally, there's also a lack of targets. These problems are interrelated, and they lead to, and they're a feedback loop upon each other, and they lead to type 1 errors, which are false positives. Unfortunately, when these false positives occur early on in development, they encourage further development of those compounds that have failed, because there's a positive, perhaps in a phase 1b study, that encourages further spending. This is a disincentive for patients to participate as well, because if they think that a study is likely to fail or a drug is likely to fail, then what's the incentive for them to participate in the study? And therefore, well, we also have a lack of reliable endpoints. I think most of you know that the primary endpoints within neuroscience are scales, and scales can tend to be subjective. And there's also the question as to whether they are clinically meaningful when they're chosen. Sometimes they're chosen because of investor preference, or that there just hasn't been any further development of endpoints within CNS. We all know about the high placebo response within CNS, and it's particularly the case in CNS because of that anticipation of treatment. And we're going to talk a little bit further about that later on in the presentation. But when there's a high placebo response, it also means that it's more difficult to separate from that placebo response. Therefore, the bar for any drug that goes into development is much higher, and that makes it more difficult. And again, that's part of the feedback loop I talked about in the first bullet. There's also poor assay sensitivity. And what does that mean? That means that there's an inability to distinguish from a less effective or a completely ineffective treatment. Without assay sensitivity, a trial is not internally validated, and therefore it's just putting it plainly, it's incapable of answering the question that is asked of it. Finally, I think we all understand that there's variability within sites and patients. We all have our go-to sites that we can trust, but there's also sites out there that perhaps are naive in research. Patients perhaps haven't participated within clinical studies before. Patients may not even understand the fact that they are participating in a clinical study, despite the processes used to bring them into the study. And therefore, that confusion between a research alliance and a therapeutic alliance gets muddied, and it makes the bar higher for seeing any results within a given study. So these all conspire to create the situation we find ourselves in today, and therefore it requires oftentimes an increased sample size. And instead of the false positives problem that we have early on in research, those larger sample sizes can lead to false negatives, and or the incentive to avoid false negatives leads to increased sample size, which of course means higher costs and therefore crowding out of perhaps other potentially more effective studies. So what can we do to improve neuroscience clinical research? Well, there's a couple of alternatives here, a couple of approaches that have been suggested here. And one is that I think we all understand that the DSM and the current version of the DSM-5, is the holy grail of clinical research within neuroscience. But all too often, it